All right, guys, welcome back to the final portion of your notes. We just talked about how our already genetically diverse combinations of chromosomes become even more genetically diverse because of crossing over. So basically, crossing over multiplies the already huge number of different gamete types, which are produced by independent assortment. So now that 6 million number is blown out of the water because it's way more genetically diverse than it already even was. Okay, so let's kind of look at a side-by-side comparison of meiosis versus mitosis. We got mitosis on the left, meiosis on the right here. So we know that mitosis, both of these begin in interphase. Mitosis goes into prophase, duplicated chromosomes condense. In metaphase, the chromosomes align down the metaphase plate. In anaphase, the sister chromatids are pulled apart through the spindle fibers, pulling them to each pole. And during telophase, each new daughter cell is separated and formed during telophase and then cytokinesis. So we have two diploid cells at the end of mitosis, two diploid cells. At the end of meiosis, let's start at the top. Prophase 1, we have kind of the same thing. Our nuclear envelope disappears. Our chromosomes condense. Metaphase 1, the tetrads line up while they do crossing over and things like that. During anaphase 1 and telophase 1, again, the spindle fibers form, pull them apart. They're split up. And now at this point, at the end of meiosis 1, we have two haploid cells. So at the end of meiosis 1, it's going to look pretty similar to mitosis. But here's where it starts to get different. Meiosis 2 is where things differ from mitosis. So we go through prophase 1, metaphase 1, anaphase 1, telophase 1, and we have our result that is just like mitosis. We shift over to prophase 2, metaphase 2, anaphase 2, telophase 2. At this point, at the end of meiosis 2, those cells are now 4 haploid daughter cells. So instead of 2 diploid daughter cells, we have 4 haploid daughter cells. And if you look closely, there's the same amount of genetic material, the same amount of chromosomes. They're just split up into 4 haploid cells instead of 2 diploid cells. That's going to be the big key to remember here. You have, in mitosis, a result of 2 diploid daughter cells. And in meiosis, you have a result of 4 haploid daughter cells. They have the same amount of chromosomes. It's just the amount of cells those chromosomes are split up into that's important. So the key difference between mitosis and meiosis is the way that the chromosomes align along the metaphase plate. So they are going to align quite differently. In mitosis, the sister chromatids line up. In meiosis 1, the homologous pairs line up. So you have the 2 pairs lining up instead of single chromosomes. Now, again, different things are going to happen in mitosis versus meiosis. DNA replication in mitosis occurs during interphase before division even begins. In meiosis, it occurs once during interphase before meiosis 1 begins, so only once. In mitosis, the number of divisions that we have 1 division, including prophase, metaphase, anaphase, and telophase. In meiosis, we have 2 division, and each set of division has its own prophase, its own metaphase, its own anaphase, its own telophase. In mitosis, when it comes to synapses of homologous chromosomes, well, this isn't even a thing. This doesn't even happen. But in meiosis, synapses is unique to meiosis. During prophase 1, the homologous chromosomes join along their length, forming tetrads, which are groups of 4 chromatids. Synapses is also associated with crossing over between non-sister chromatids. Now, when it comes to the number of daughter cells and genetic composition, in mitosis, you have 2 diploid, genetically identical to the parent, daughter cells. In meiosis, you have 4 haploid cells, daughter cells, that contain half as many chromosomes as the parent cell. So they're not genetically identical to the parent or each other. Also, what role does mitosis play versus meiosis play in an animal body? Well, mitosis enables the multicellular adult to arise from a zygote. It produces cells for growth and tissue repair. Now, meiosis produces gametes, the sex cells. It produces sperm in males, eggs in females, and it reduces the chromosome number by half and introduces genetic variability among the gametes. So this is how, in animals, this is how our genetic variations occur. This is how we are not identical to our parents. We are not identical to the human sitting next to you. Because of this diversity from meiosis, we're different. We have different genes, and that's a good thing. Okay, another thing to look at here, whether a person or an organism is genetically a male or genetically a female, the Y chromosome is going to decide. We have this X chromosome, the larger X chromosome, and this tiny little Y chromosome. If you are a biological male, you are going to have an X and a Y chromosome. If you are a biological female, you are going to have an X and an X chromosome, so 2 of these X chromosomes. So the mother begins with XX chromosomes, the father begins with XY chromosomes. So right there, that tells you the father is the parent that determines whether you are a male or female. So that can only come about from the father. Now, we have meiosis 1 occurring, meiosis 2 occurring, then we have fertilization, and depending on if the father's X chromosome is dominant or the father's Y chromosome is dominant, that's going to determine whether the new offspring is a male or a female. If the father's X chromosome becomes dominant in this fertilization process, you will have 2 X's and you will have a daughter, because the mother only provides X's. If the Y chromosome from the father is dominant, well, then you will have XY, and XY is a son, a biological male. So again, the Y chromosome decides whether an organism is biologically male or biologically female. Sometimes there are errors in meiosis and things cross over incorrectly. Our chromosome pairs don't exactly line up the way they're supposed to. Something such as Down syndrome is a problem. If you look down here, each homologous pair is supposed to have just that, a pair, 2 chromosomes. But down here on the 21st chromosome, there is an extra chromosome. And when we have that extra chromosome on the 21st chromosome, that's how Down syndrome occurs. Some people call it trisomy 21 because there's a tri or a third chromosome on the 21st pair. And that causes Down syndrome to occur. Another question is this particular person male or female? Well, if you look down here at the 22nd, those appear to be the same. And if they are the same, XX, you have a female. If they were different, XY, we would know that it was a male. Okay, there could be other errors in fertilization. Should an egg with an extra chromosome be fertilized and then the offspring have an extra chromosome? So if it gets fertilized with an extra chromosome, that's going to cause some problems. In humans, this often occurs with the 21st pair. So again, this extra chromosome comes in, and now it's attaching itself on the 21st pair of chromosome, and now you have Down syndrome. That is a problem. So when it comes to this whole crossing over and differentiating all of your chromosomes, this can be a pretty tough thing. There's no way to determine what the results are going to be, but because of this variation, we have such genetically diverse species. Different organisms are different from the next because of this difference here. All right, guys, that concludes our notes. Finish these up and turn them in.